Santham Academy. Welcome to Digital Kaksha. To get updates on new videos, click the subscribe button and the bell icon.
Good morning all of you. So, today in this video, we will be studying about the post-fertilization events. After the fertilization, that is the double fertilization which occurs only in the angiosperms, which results in the formation of zygote and endosperm. So, after this fertilization, the post-fertilization events are there. So, let us see what are the post-fertilization events. So, the post-fertilization events comprises of 4 sub-events. Okay. Number 1 is the development of the endosperm. Number 2 is the development of embryo. Number 3 is formation of fruit from the ovary, and number 4 is formation of seeds from the ovules. Okay, so there are 4 sub-events in the post-fertilization events, that is number 1, development of endosperm, development of the embryo, formation of fruits and formation of seeds. So, we will take these sub-events one by one. So, starting with the first post-fertilization event, that is development of endosperm. So, you all know that endosperm, which is formed as a result of double fertilization, it is a triploid tissue which gives nourishment to the developing embryo. So, always the formation of endosperm precedes the development of embryo. You must have understood why this occurs, because the endosperm have to provide nutrition to the developing embryo. So, the development of endosperm precedes the development of the embryo. So, the endosperm is being formed in the central cell, as the triple fusion occurs in the central cell. Now, that primary endosperm nucleus inside the central cell, and the central cell gets converted into primary endosperm cell. Now, this primary endosperm cell will continuously undergo cell divisions, that is mitosis cell division, resulting in a mass of cells called as the triploid endosperm tissue. PEN gets converted into PEC, that means the central cell gets converted into primary endosperm cell, and the primary endosperm cell, it continuously divides to form mass of cells called as triploid endosperm tissue. The way this division occurs is little different. What happens? First, continuous nuclear division occurs. First, continuous nuclear division occurs without formation of the cell wall, and after a mass of nucleus is being formed, then only the cell wall formation will take place. So, first free nuclear divisions will occur and later the cell wall formation will take place. Now, we will take example of coconut. You all must have seen the coconut, you must have consumed the white part of the coconut, and you must have enjoyed the coconut water also. So, what is this coconut water and what is this white part of the coconut which you consume? The coconut water is actually the free nuclear endosperm. Free nuclear endosperm means that the primary endosperm cell has divided, that means the nucleus has continuously divided without the formation of cell wall. After that, the cell wall formation occurs. So, the coconut water is the free endosperm nuclei, and the white part of the coconut which you consume, which is edible, that part is the complete endosperm in which the cell wall has been formed. So in this way you can differentiate the free nuclei endosperm and the cellular endosperm. The white part is the cellular endosperm and the coconut water is the free nuclear endosperm. So what happens? The cell wall formation here is occurring from the periphery towards the center. After some time you will see that the coconut water has disappeared and completely the whole cellular endosperm has been formed. So this was how the endosperm is being formed, which precedes the embryo development. Now we will move to the development of embryo. After a sufficient amount of endosperm is being formed, then the development of embryo starts. You know that zygote is also being formed as a result of syngamy, that is the first fertilization which have occurred. So that zygote is a single celled structure. So zygote is a single cell structure and continuous mitosis division occurs in the zygote, resulting in formation of embryo. Now, how you can define this embryo? Embryo, it is an organized mass of cells which has the potentiality to form a new plant. In case of plants, we can define the embryo as an organized mass of cells which has the capacity or which has the potentiality to form a complete new plant. So that is an embryo, and this complete stages in the formation of embryo that is studied under embryogeny. What is embryogeny? The study of the stages in the formation of embryo, that is called as embryogeny. So here we will be discussing about the steps in embryo formation in a plant. So let us see how this embryo formation occurs. So starting with the single celled zygote. The single celled zygote, that you know that it is diploid in nature. The single cell zygote is diploid in nature and this zygote will undergo mitosis division. So the single cell zygote will undergo mitosis cell division to form two daughter cells. You all know that as a result of mitosis, two daughter cells are formed. But here in this case, asymmetric mitosis division occur. That means it results in formation of two daughter cells, and these two daughter cells, they differ in size. That means one large cell is being formed and one small cell is being formed, and this is the two-celled stage. Okay, so the large cell, it is called as the basal cell, and the small cell is called as the embryonal cell. As the name indicates, embryonal cell means that that particular cell is going to divide continuously to form the embryo. So zygote divides by mitosis to form two unequal cells, that is the basal cell and the embryonal cell. Now let us see what is the function of this basal cell. So the basal cell will continuously divide by mitosis cell division to form a chain of cells, and this chain of cells is called as the suspensor cells. What happens? First, the basal cell will continuously divide by mitosis cell division to form a chain of cells. As you can see in the diagram, a chain of cells are being formed, and this chain of cells are called as the suspensor cells, and these suspensor cells, they hold the embryonal cell. By the time the suspensor cells are being formed, the embryo will also divide, but the process will be slow as compared to the formation of suspensor cells. Once the suspensor cells have been formed, then the division in the embryonal cell will be faster. So as the suspensor cells are being formed, then you can see at the base the embryonal cell has also divided to form a mass of cells. This mass of cells is called as the globular embryo. As it is spherical in structure, so it is called as the globular embryo stage. So, from the suspensor cells, the globular embryo hangs. Now, let us see what is the function of this suspensor. Why this suspensor has been formed? The function of this suspensor is to push the embryo towards the endosperm. So, these suspensor cells are present towards the micropylar end and this embryonal cell is present towards the central cell. As this suspensor is being formed, the embryo is pushed towards the endosperm. So, here we have reached the globular embryo stage, and after the globular embryo stage, here an invagination occurs. Invagination means that a formation of furrow occurs towards the center. The formation of furrow occurs towards the center, which results in this type of structure. As you all can see in the diagram, it is the shape of a heart. So, the globular embryo, it attains a heart-shaped structure, and this stage is called as heart-shaped stage of the embryo. So, from the globular embryo, the heart-shaped embryo is being formed. Now, why this invagination occurs or why this furrow formation occurs? It occurs for the formation of the cotyledons in case of dicot plants. Okay, so in dicot plants, as you know that the seeds have got two cotyledons. So for the formation of two cotyledons, a furrow formation or an invagination, it occurs towards the center, resulting in a heart-shaped embryo. So, is it clear? Now we will move to the next stage, that is the mature embryo stage. So, this mature embryo stage, here you can see that the cotyledons, they are completely formed. The cotyledons are completely formed, and towards here a mass of cells are also being formed. Still they are being suspended by the suspensor cells. Now one thing which is being labeled over here, the last cell of the suspensor, that is called as the hypophysis. Here also the last cell is there, which is called as the hypophysis. Now we will see what is the function of this hypophysis. Here you can see two cotyledons are there, and here near to the suspensor cell, the part which is there, that is called as the radicle. You all know that radicle is that part of the embryo which is going to form the root. And here you can see towards the cotyledons, you can see plumule is there. Plumule is that part of the embryo which is going to form the shoot. So here you can see two cotyledons are there, and towards the suspensor the radicle is there which is going to form the root, and away from the suspensor the plumule is there which is going to form the shoot. Now here you know that the radicle, it is covered by a root cap, or you can say that the primary root is always covered by a root cap which protects the root when it enters or moves along the soil. So that protection is being provided by the root cap. And the hypophysis, which is the last cell of the suspensor, that hypophysis forms the root cap. Okay, so that is the function of hypophysis, which is the last cell of suspensor to form the root cap. So here we have reached the mature embryo stage, which is also called as the torpedo stage. So once more we will see the stages. Single celled zygote is there, diploid in nature. You know that when the zygote continuously divides by mitosis cell division, all the daughter cells which are being formed, they all will be diploid in nature. So the zygote will divide by mitosis, that will be asymmetric mitosis, resulting in formation of two unequal cells, the bigger basal cell and the smaller embryonal cell. The basal cell will form the suspensor and the embryonal cell will form the embryo. So the division will occur. The basal cell will divide to form the suspensor cells, which is a linear chain of cells, and the embryonal cell, it will form the globular embryo by continuous divisions. This is called as the globular embryo stage. Now, in the globular embryo stage, slowly an invagination occurs or a furrow formation occurs, which results in heart-shaped embryo, and it is called as heart-shaped embryo stage. This furrow formation is important for the formation of cotyledons. So, here the two cotyledons are being formed, and the upper part, which is near to the suspensor, that forms the radicle, and the part which is away from the suspensor, that forms the plumule. So in this way, the radicle, the plumule will be formed, the cotyledons are being formed, and the last cell of the suspensor, it is called as hypophysis, which will be forming the root cap. So this was about the development of endosperm and the development of embryo. Now we will study the structure of monocot embryo. As we have studied the stages in formation of a dicot embryo, the same structure is that of a dicot embryo, which we have studied in the mature embryo stage, consisting of two cotyledons, the radicle, the plumule, and the suspensor cells. Later the suspensor cells, they degenerate. So now coming to the structure of monocot embryo. As you know that the monocot embryo, it has got a single cotyledon. So a single cotyledon is there in case of monocot embryo, and two cotyledons are there in case of dicot embryo. Now let us see the structure. So this is the structure of a monocot embryo. Here you can see one structure labeled over here, that is called as scutellum. Scutellum is the other name for the single cotyledon. So in case of monocots, mainly the grass family, the cotyledon is called as scutellum. So we can use scutellum and cotyledon as alternate words in case of monocot, while in case of dicot the term cotyledons are being used. So scutellum, that means the single cotyledon present. Now here you can see this entire structure. This part is called as the epicotyl. Epicotyl means that the part which emerges above the cotyledon, that is called as epicotyl, and the part which emerges below the cotyledon, that is called as hypocotyl. So here you can see that epicotyl is there, and epicotyl has got the shoot apex. Shoot apex simply means the plumule. Shoot apex means the plumule, the part which is going to form the shoot of the monocot plant. So that is the shoot apex or the plumule. Now here the shoot apex or plumule is covered by a covering, and that covering is called as coleoptile. What is coleoptile? The covering which protects the shoot apex or which covers the shoot apex or plumule, that is called as coleoptile. So this coleoptile and shoot apex together make the epicotyl. Now moving to down, we can see hypocotyl is there. Hypocotyl means the part which is below the cotyledons, that is hypocotyl. So here you can see that below the cotyledons the radical part emerges and this radical will form the root. So here you can see the radical is there and the radical is being covered or protected by a root cap, and here again a covering is there which protects the radical and the root cap, that is called as coleorhiza. So here the protective coverings in case of shoot is coleoptile and in case of root it is coleorhiza. So this coleoptile and coleorhiza are present only in monocot embryo. So that is absent in dicot embryo. One structure here you can see, that is epiblast, a small finger-like structure, that is the epiblast. So what is this epiblast? It is nothing, it is simply the residual second cotyledon, or we can say that here as single cotyledon is there, the residue of the second cotyledon is called as the epiblast, which is not a fully formed cotyledon. This was the structure of the monocot embryo. Now we will move to the structure of the dicot embryo and the dicot seed. You know that the dicot embryo and dicot seed has got two cotyledons. In the structure of the dicot embryo we have already seen that. Now here we will be seeing the structure of the dicot seed. So you can see that when you open a dicot seed, two parts you will get. So these two parts are the cotyledons. These cotyledons have got the function to nourish the embryo. Endosperm also have the function to nourish the embryo, but when the initial embryo stage is there, at that time the nourishment is being provided by the endosperm. Later the nourishment is being provided by the cotyledons. After, we can say that after the endosperm is being consumed, the cotyledons, they provide the nourishment. Now after the cotyledons you can see that the seed, it is protected by two coverings. Okay, the seed, it is protected by two coverings. The outermost covering is called as the testa and the inner covering is called as the tegmen. So a seed is being protected by two coverings, the outer testa and the inner tegmen, and together the testa and tegmen is called as the seed coat. So the seed is protected by the seed coat, which is formed of the outer testa and the inner tegmen. At the center of the cotyledons, embryonal axis is there. Above the embryonal axis, the epicotyl is there, means the parts which will arise above the cotyledons, that above region is called as the epicotyl, and the part which emerges below, the part which is below the cotyledons, that is called as the hypocotyl. As you already know that the hypocotyl region will consist of the radical which is going to form the root, while the epicotyl region will consist of the plumule which is going to form the shoot. So once again we will see the parts: seed covered by seed coat having testa and tegmen. In the center the embryonal axis is there, and above the embryonal axis the part which emerges below the cotyledon, that is called as the hypocotyl. The radical will be formed in the hypocotyl region, while the plumule will be formed in the epicotyl region. Radicle will form the root and plumule will form the shoot. Now one more thing I want to tell you, that we have studied in the structure of the ovule that the ovule, it is attached to the placenta by a stalk-like structure called as the funicle, and the region where the main ovule gets attached to the funicle, that region is called as helium. I think all of you must have seen the gram seed. At the base of the gram seed you can see a scar, and that scar is called as the helium. That scar is the region where the funicle was attached to the ovule. Okay, so that is called as the helium, and that scar represents that the funicle was attached to that particular ovule at that region. Now just above that you can see a small opening in the gram seeds. When you put that in water, when they swell up, you can see a small opening at the base, and that opening is called as the micropyle, which we have already studied in the structure of the ovule. So that small opening is micropyle, and the function of that micropyle here in the seed is that from the micropyle the water for the germination is taken in. The seed takes the water for the germination process through the micropyle. It also takes the oxygen from the same pore. So what is the function of micropyle? Number one is that it allows the entry of the pollen tube, that is before the fertilization process. After the fertilization, when the seed formation has occurred, the water and oxygen, it enters the seed through the micropylar region. So this helium and this micropyle is visible in a normal dicot seed easily. So in this class we have studied about the formation of the endosperm, the formation of the embryo, that means the different stages in the formation of embryo, the two-celled stage, then the globular embryo stage, after that the heart-shaped embryo, and then the mature embryo. Then we have studied about the structure of the monocot embryo and how the dicot embryo differs from the monocot embryo. Then we have studied about the structure of the dicot seed, different parts of the dicot seed, and when the seed germinates, how it will get the water and oxygen. I hope whatever I have told in the class, that is very much clear to you. Rest of the topics we will deal in the next class. Thank you. Take care.
Thanks for watching. If you like this video, make sure to like, share and subscribe for more.